on stem cells and the science behind it, and he chose to say yes to my invitation for a smaller group. But you know, I think that it's a smaller group and it's very intense, right? You're go-getters, and I know that you're going places, and I feel extremely privileged to have him as a guest. So please put your hands together for Scientific Advisory Board member, Mr. Christian Drapeau.
Thank you so much. Thank you. It's a real pleasure to be here, first and foremost because I'm from here. So it's always pleasant to come home. So anytime, Martine, you invite me, I will say yes. It's a little strange, though, to speak English while being here, but I guess we will do. You will all speak French next time, right? Okay, so I'm here to talk to you about Blu. Blu is all about stem cells, and to understand really how could I say, the importance, let's say, of what we're talking about. In the world of science, scientists research stem cells because there is this deep understanding that adult stem cells have enormous regenerative potential. This is why there's so much excitement, so much work being done with adult stem cells. The question that is never asked is that stem cells are already in your bone marrow. They are in your blood, they are in all your tissues, they're already in your body since the day you are born. When you take them out, tweak them in the lab and re-inject them, and this has, you know, great interest in the medical world, the stem cells are not different than the stem cells that are already in your body. So to me it begs a very important question, and essentially that's what we'll be talking about today. What is the natural role of stem cells in your body? Because when we understand this, then I think it is called to change not only the way that we understand health and wellness, but also the way that we practice medicine. So I will be using a lot of technical information today. I just want to be clear. Understand here that you don't have to remember any of this. It is not really important for you to know this information. The only reason why I'm sharing it is because if you're not aware of this information, it will appear that I'm making pretty substantial and incredible claim about what stem cells can do. So I just want to make it clear to you that this is coming from real scientific research, not just one or two studies, but hundreds of studies. I'm just picking a few of them that illustrate the whole concept in a way that will make it very clear. If at some point the information appears too technical, don't worry, I will summarize everything in very simple conclusions. The message itself is simple. I just want you to really see that it's based on real data. So far so good?
Yes.
Okay, so let's start. So first we need to define what are stem cells. The best way to define what is a stem cell is to first define what is not a stem cell. All the cells in your bodies are called somatic cells. These are cells that are specialized. They will do one specific thing in the body. They will never transform. A cell of your muscle is always going to be a cell of your muscle. A cell of your heart will always be a cell of your heart. They'll never transform into anything else, and for the most part, they will not multiply. If you go to the gym, you work out very hard, you lift very heavy weights, you will not have a cell of your skin see the muscle below and just say, gee, the muscle needs some help, so I'm going to go and become a muscle cell. It won't happen. Cells are one thing and they stay that way. At the other end of the spectrum, you have stem cells. Stem cells in your body, and understand what I'm saying here, stem cells are nothing, meaning like they have no function. A stem cell does absolutely nothing in your body. Its function is in its ability to transform into another type of cell. Historically, traditionally, if you go to med school, even today, stem cells will be, you will be told that the function of a stem cell is to transform into blood cells. They are essentially the precursors to white blood cells, red blood cells, and platelets. What has prompted all the development that we'll be talking about today is that in the early 2000s, scientists started to observe that stem cells could become other things than just blood cells. It was a huge breakthrough discovery, and it happened through studies like, for example, the first study that I came across that really triggered all my work into this field was an article that a colleague sent to me. The title of the article was Turning Blood into Brain. It was a study on leukemia in which scientists irradiated female mice, so it kills all the stem cells, and they tested a treatment for leukemia, which was irradiation and then transplant of bone marrow stem cells. So they basically transplanted male stem cells into female mice because we can follow the Y chromosome. So through this procedure, then scientists could see that, yeah, you can reconstitute the bone marrow, you can reconstitute the blood, but a few weeks later they found a brain cell with the Y chromosome. So it can only come from the bone marrow. So those kinds of observations started to be done early in the process. So here's one study that I like to use to illustrate this process of transformation of stem cells into tissue cells, because I could use hundreds of studies, but that one illustrates the phenomenon in a way that allows us to answer indirectly another question that I think is very relevant, which you may have in your mind, or if not, maybe it'll be there tomorrow in a week from now, which is the fact that we now go to Mars. We can film a living brain. We can put tiny cameras in your bloodstream to film your bloodstream. We have a level of scientific sophistication. And here, at this time in our history, we discovered that stem cells are your repair system. And it's been like this forever, and we've never seen that before. How is it? How is it that something like this was missed? And that study kind of gives us an indirect answer. So here's a study in which scientists took a double beaker, so a small beaker sitting into a bigger beaker. And the top beaker, the membrane here at the bottom, is made of a semi-permeable membrane. So it has holes big enough to allow the passage of molecules, but small enough to prevent the passage of cells. So cells cannot go through. So you put stem cells here, they stay in that compartment. They put at the bottom here a piece of damaged liver. These liver cells stay in that compartment. But any kind of SOS molecule released by a damaged tissue can travel and touch these stem cells. And within 8 hours, these stem cells started to transform into liver cell. Now, the way in science to identify a cell, until fairly recently, methods have changed, but until recently, is essentially pathology. You just put a piece of tissue or cells under the microscope, and given their shapes, the number of nuclei, various visible characteristics, you can tell this is a liver cell. But it's impossible to tell, yesterday it was a stem cell. So the process is completely invisible. It is one thing one day, the day after it's something else, and you've lost the complete transition until a day when you can tag that stem cells and then follow the tag. Then you can visualize. It's a little bit like, I'll make an analogy, but a few years ago I was watching a documentary on sharks on the West Coast. Sorry, on the East Coast. Well, right here. And but it was done in Florida. So scientists assumed for a long time that sharks are not always by the shoreline. They come in a cyclic manner. And the idea is that they go further in the ocean and then they come back to the shore. So one day they developed a tool called the GPS and they tagged the shark. And they discovered that no, it disappears from Florida because it swims up the coast up to fairly north, like Newfoundland, and then come back down. When you start to tag something, you start to discover the behavior, you discover behaviors that you did not expect before. That's the same thing here. So following these studies, the tool here is a very interesting tool called green fluorescent protein. It is a protein extracted from a jellyfish. It's a spontaneously fluorescent protein. So you just shed light on it and it glows. If you went into the ocean when you have these, you may have seen them at night. So it's a protein. Given that it's a protein, you can derive the DNA. Now if you derive the DNA for this protein, you can inject that DNA in the nucleus of a stem cell. So now that stem cells will produce this protein, so it will be green. But if that stem cell multiply and becomes a population of cells that transform into liver cell, as an example, You look at them under the microscope, you can tell they're liver cell, but the way to tell that they were a stem cell the day before, you just shed light on them and they'll glow. So now you can visualize the whole process. So the study is very simple. Scientists took mice, they irradiated them, so it kills all the stem cells. They injected fluorescent stem cells, triggered an injury to a tissue, and then they observed the process of tissue repair. And here's what it reveals. If you damage a muscle in a mouse, 6 weeks later, the muscle is completely reconstituted. By damaging, I mean they injected a toxin that completely destroyed the muscle. The leg was paralyzed, no movement. 6 weeks later, movement is recovered, but the muscle is entirely green, derived from the bone marrow. Heart attack, or damage to the heart, same thing. It's filled with new heart cells that are green. Same thing in the skin after a burn. Same thing in the kidney. Same thing in the lung. Same thing in the pancreas. I'll come back to that later. This is a pancreatic cells producing insulin totally normally, naturally, but it comes from the bone marrow. So the point here, I'm just showing a few studies to show while stem cells have been traditionally believed to be precursors only to blood cells, in the early 2000s, a discovery was made that it's not true. Yes, they do become blood cells, but they can also become cells of various tissues. With these early observations, at the beginning, the early observation, 2000, 2001, was the observation that stem cells could become brain cells, heart cells, and liver cells. So when that information comes out, you can take one of two positions. Very conservative positions would be, these were bizarre stem cells. Another more aggressive position could be to say, man, we've just discovered a new role of stem cells. It's like you observing butterflies and caterpillars, and you've never seen that a caterpillar becomes a butterfly. Who would ever imagine that a butterfly comes from a caterpillar? Until you see one. Then you can conclude two things. That was a weird caterpillar, or you can say, well, let's test them. Maybe butterflies come from caterpillar. So I'm saying this because we decided to take a more aggressive posture, and on the basis of that information, we published an article in 2002 in the journal called Medical Hypotheses, in which we simply proposed it looks like stem cells constitute the natural repair system of the body. It was just an idea at the time, a very daring idea. Today, what I'm going to show you looks so obvious. It is so obvious that it's almost like difficult to fathom that just 10, 15 years ago, we were laughed at with something like this. I remember speaking into a conference with doctors or scientists, and one day this older fellow stood up and he said, son, I have to tell you something because it looks like nobody is telling it to you. You're just an idiot. And it's fine. I'm just saying this because I want to put all of this into not the context of saying they're stupid because they don't see it. It's more just to say it is the natural progression of scientific information. If I was not told in college that stem cells can do this, I won't believe somebody who talks about it, that does not come from academia. So I just want to explain, this is just the natural process of scientific information. It's discovered, it takes 1 or 2 decades before it slowly sinks in, and then it becomes part of academia. But there's a lot of science behind this. So let's look at what that process looks like. What I'm going to do here, I'm going to summarize the whole process. I wrote a book about this whole concept. There's more than 400 scientific references in the book. Just understand here, this is not an idea anymore. This is very, very solid. Anytime there is an injury in a tissue or a problem, a lot of these studies have been done. And also, another parenthesis that is important. I will mention many times disease name. I want to be very clear. I am not in any way, shape, or form implying that Blu will have an effect on these diseases. All I'm saying here, I want to educate you about what stem cells are doing, but in the world of medical science, we very rarely study healthy people. If you take 50 healthy people, you give them a carrot every day, and 6 months later they're still healthy, you can't conclude that carrots are good for you. You need to start with a problem where you can quantify a change in order to show that something is good. And at the end, your conclusion can be strictly about health, but the science available to understand the process is all coming from diseases. So that's why I'm mentioning diseases. And it's also because I want to properly report the information and the studies in the scientific literature. So a lot of these studies were done with heart attack, broken bones, stroke, burn to the skin. So within hours after an injury, compounds start to show up in the blood that have been very well documented today. And they reach the bone marrow, and they're compounds very, very well known in medicine. They are virtually daily molecules being used in the whole field of oncology, cancer research, cancer treatment. So they reach the bone marrow, they trigger the release of stem cells from the bone marrow, or the detachment of stem cells from the bone marrow matrix, and then the stem cells gets into the blood circulation. So within about 3 to 5 days, the number of stem cells in circulation start to increase, and will increase about 3 to 10-fold. So we all have a baseline number of stem cells in circulation. Within 3 to 5 days after an injury, this will increase 3 to 10-fold. The number of stem cells that we have is like the number of red blood cells, the number of white blood cells, platelets. It's different for everybody. We have a healthy range that has been established for almost all blood parameters. No healthy range has been established yet for stem cells. That's what I'm working a lot right now through a review of the scientific literature, because the conclusion of everything that I'll be talking about today is that there is a link between how many stem cells you have in your bloodstream and your ability to be healthy. Below a certain number, your body is at risk for developing problems. Right now it's a free statement that I make. I'll demonstrate it in the rest of this lecture. But my point here is that the range between what we can find in the blood of various individuals is fairly broad. I'll come back to that. Okay, so now stem cells are circulating everywhere in the body. They don't know where to go. They don't know what is the tissue that is in need of repair. So how do they find what is the tissue in need of repair? As they circulate, so within about 2 days after the injury, the tissue, the affected tissue, will release locally another compound, again, very well known in science. It's called SDF1. Very locally, that compound is released, so the stem cells circulate everywhere in the body, but when it goes into the fine capillaries of the affected tissue, right there, locally, these compounds touch the stem cell, and it triggers the migration of stem cells out of the blood into the tissue. When the stem cell arrives in the tissue, it will keep seeing where this molecule is being released. The stem cell will crawl to the site of the injury, and upon contact with cellular debris of the tissue, they will transform, just like in the beaker before with the liver cell, they will transform into cells of that tissue. It is your repair system. Isn't it beautiful?
Yes.
Think about it. Let's not make this super complex here. You have an immune system. Nobody would question that it's an important system in the body. You cut yourself, you have a bacteria that goes into your cut, you have an infection, so you will release specific immune cells from specific tissue called your lymphoid tissue. The immune cells will go everywhere in the body, but they will reach, where will they go? Ultimately, right there in your cut. They will go right beside the bacteria, and through a very well orchestrated phenomenon, they will kill the bacteria. It's your immune system. It's what it does. It's well coordinated to do that. For a stem cell to leave the bone marrow and go exactly where you have an injury, it will be like me telling you, you know what, I know somebody in Beijing, just go randomly walk in Beijing and meet that person. Impossible. It needs to be coordinated. So we have here a specific tissue, your bone marrow, that releases a specific cell, your stem cell, that will go according to a very coordinated process to the site of an injury and will repair the tissue. It is, without any stretch of the imagination, your repair system. So the body has a repair system. And in this repair system, you have the release of stem cells from the bone marrow, there's circulation in the bloodstream. These are the various steps. There's circulation in the bloodstream, their migration into tissue, and then their proliferation or multiplication, and then transformation into cells of these tissues. So I won't go through all these processes, but I'm showing all of this just to emphasize here that in science, you can only observe the things for which you have a tool to study. You can't discover bacteria until you've developed the microscope. You can't study sharks' migration pattern until you have the GPS. So you can't study these, you can't study all these different steps. This clicker is on Wi-Fi and there's a delay between the two. So sometimes I click and I'm not sure. There you go. Okay, so we can study all of these, but what we can study the most is the release of stem cells from the bone marrow, because we know these molecules released by an injury. If you inject these in an animal, you trigger the release of stem cells, which means you increase the number of circulating stem cells, and then you can study what is the impact between releasing stem cells and health. So the bulk of the science is about the link between the number of stem cells in circulation and health. So far, so good?
Yes.
Okay. So if I summarize what you will find in scientific literature, here are some of the key conclusions. Number one, more stem cells in circulation means that more stem cells are available to participate to the natural process of tissue repair, which means you've got two individuals with the same broken bone. The one that has more stem cells, remember we all have a different number of stem cells in circulation, those that have more are those that will repair faster and better. It's that simple. Here's one study, to illustrate this phenomenon. Here's a study that was done published in the New England Journal of Medicine, and what it did is that it investigated 500 individuals at risk for cardiovascular problem, in which they quantified the number of stem cells naturally present in their bloodstream. Then they monitored for 1 year the occurrence of any kind of cardiovascular problem, heart attack, arrhythmia, angina, whatever. And then a year later, they correlated the number of stem cells in circulation and the occurrence of a cardiovascular problems. In other words, do I see more problem in people with fewer stem cells? That was the question. When you look at the data, so all these people were separated into 3 equal groups: people with a lot of stem cells, people with average number of stem cells, people with a low level of stem cells. And then this graph shows survival over time without cardiovascular problem. Anytime you see a line going down, this is one person having a cardiovascular problem. So clearly, you see the conclusion here. The number of stem cells in circulation was one of the best predictor of cardiovascular health. Conclusion of that study. You can see people with a lot of stem cells had very, very few problems compared to people with fewer stem cells. That same relationship exists with a lot of different problems. I'm showing you one of them. Which begs then the question, if the people with, because what is then later, this was published in 2005, later it was established that the mechanism of action, or what's happening in the body behind this study, is that when you have a lack of oxygen in the heart, which is the basic of all the problems that you have in your heart, when you have ischemia, lack of oxygen, the heart will release many compounds, but 2 that are very relevant here. One is called SDF1, the compound that will attract stem cells in circulation to the heart, and another one called VEGF, vascular endothelial growth factor. The names are irrelevant, but it will make these new stem cells transform into new blood vessels. So you bring back new blood, you remove the lack of oxygen, the tissue is working better, so you don't develop the problem. It's so simple. So the same information here exists for various kinds of problems. But it begs the question, if having more stem cells mean that you can repair better, what if I can trigger the release of stem cells from the bone marrow and increase the number of circulating stem cells? Could I enhance the ability of an individual to repair? Fair enough? Okay. So here, to answer the question, I just want to position all of this into the greater context of what you already know about stem cells from what you've read in the media. I don't want to compare what I'm going to talk about to stem cell treatments, but I want to make sure that you link what I'm telling you with what you already know about stem cells. When we talk about stem cell treatment, we talk about the isolation of stem cells from a source, which can be an umbilical cord, your own blood, your fat tissue, your skin, your dental pulp. There are many sources of stem cells, but none of them will give you enough stem cells for a robust injection of stem cells. So scientists have developed ways to multiply the number of stem cells. Then you inject them in the bloodstream. So what we have done is that we've taken stem cells, for example, from the blood, we multiply them, and we re-inject them in the bloodstream. So what we have done is that we have increased the number of stem cells in circulation through an injection. But once we discovered that we release them all the time, it's a natural process of the body, then it begs the question, what if I release my own stem cells? What if I increase the number of stem cells in circulation simply by supporting this natural process of releasing my own stem cells? And that's what is meant by endogenous stem cell mobilization. It means the release of your own stem cell. So just to put that in the context here, I'm not talking about something that is so far out of what we know of stem cells. I'm talking about something that is very mainstream. We're just increasing the number by using a different strategy, releasing your own stem cells. So let's look at what we have in science. Again, I'm picking studies. It may get complicated, but I just want to expose you to that information. The conclusion is very simple. Here's a study that was done by the National Institute of Health in America. So they took mice, they triggered heart attack by making a knot in the coronary artery, so they deprive blood flow. Then they released the knot, so the animals have a heart attack, then they let them recover, and then they separated them in two groups. One group is control, the other group they inject those compounds known to dramatically increase the number of stem cells in circulation. So the difference is simply that the treated group had more stem cells in circulation. Then they looked at these mice 27 days later. If you look at the control group, you can see fairly severe scar tissue, no new blood vessels, 17% survival, and poor cardiovascular function. Now look at that ventricular wall here. In the experimental group, complete renewal of the ventricular wall, functional new blood vessels, 73% survival compared to 17, and quasi-normalized cardiovascular function. So in a world where there's nothing we can do to recover from a heart attack, here's a 18-year-old study that shows that you can virtually completely recover in a month. The reason why this is not used in humans is simply that the protocol, as it was used in these mice, can have severe side effects in humans. So that cannot be used as such. It would be like telling you, well, we'll help you recover from your heart attack, but we can give you a stroke. It's not a great marketing message. So they're working on drugs like this that are safer without these side effects, but the problem is not linked to stem cells, it's linked to the drug itself. So the study here is extremely relevant because it tells us one very interesting message, is that if you can increase the number of stem cells in circulation, the body does have the ability, the machinery, the biology to use these stem cells for tissue repair. Here's another one with the endocrine system. Let's say diabetes. To show that we can have an effect on something like diabetes or functioning of the endocrine system, I need to demonstrate two things. I need to demonstrate that stem cells can naturally, on their own, leave the bone marrow, go to the pancreas, and become insulin-producing cells in the pancreas. If I can show that this is a natural process, then the next question is, if I enhance that natural process, can I enhance it enough so that it changes the course or improves a condition like diabetes? Fair enough?
Yes.
Okay. Here's the first study. Ingenious studies. Sometimes scientists develop protocols, and it's really ingenious. So scientists took male mice in which they coupled the gene for insulin to the gene for green fluorescent protein. So you take the very first cell that will become that male mouse, so the first embryonic fertilized egg, and in that cell you make a genetic manipulation. The gene for insulin, you attach it to the gene for green fluorescent protein. So in that animal, any cell that starts to make insulin will turn
Green.
very good, will turn green. One person is listening. Okay. So now they irradiated female mice and they injected stem cells from these male mice into these female mice. So I have female mice whose entire body is made of cells that have the double X chromosome, no Y chromosome, except the bone marrow is made of stem cells with the Y chromosome, and if one of these cells starts to make insulin, it turns green. So they isolated cells from the pancreas a few weeks later, I think about 6 weeks later, and if you look at that specific cell, and you shed a light on it, it starts to glow. So it has transcribed the gene for insulin. This is from the pancreas. Now the question is, it has transcribed the gene, but is it a normal cell that is normally making insulin? By using a red tag for insulin, that cell is actually making insulin. And it's actually making insulin right between these other two normal cells. Oops, sorry. Did I do something wrong? I must. Okay, so just to show here that it's a very normal cell. It's not an overactive, it's not a sluggish cell, it's not a Frankenstein cell, it's not a bizarre cell that comes from the experiment. It's a very normal cell of the pancreas. And at the end, when I'm done with the presentation, what you will understand is that that cell is like every single cell in our pancreas today. At the age that we all are in this room, we don't have one cell in our pancreas that we're born with. They've all been replaced many times over through this very natural process of going from the bone marrow, going to the pancreas, and making insulin in the pancreas. Now, next, oh, and then it is a cell that comes from the bone marrow because it has the Y chromosome. So stem cells can on their own leave the bone marrow, go to the pancreas, and become an insulin-producing cell. If you've studied biology, I know right now you're basically screaming while I don't hear you because it makes no sense. This is not what I was told when I did my pre-med school. It's not what happened. But the data is very, very clear. So next step. To study now if it could have an effect on the course of the development of diabetes, we need to understand that in science, we study disease on animals because we obviously cannot use humans, at least in the early stage of developing drugs or remedies. So we need to study them in animals. I dare you to find a mouse that has diabetes. I dare you to find an Alzheimer's mouse. A mouse that forgot what a cat is, you won't find one. I dare you to find a mouse with multiple sclerosis or Parkinson or any of those. They don't exist. You need to find a way to trigger the problem in the animal or a problem that resembles the human disease, and then you try to do something about it. So Alzheimer's, for example, you make a hole in the skull, you sprinkle some aluminum, and you will see in the brain the same lesions that you would normally see in a human that has Alzheimer's. In diabetes, it's a toxin called streptozotocin. It's a bacterial toxin. The name is irrelevant. And so here is a normal, islet of Langerhans, which is the part of the pancreas that makes insulin. And 2 days after injection of streptozotocin, this looks like this. It's completely destroyed. These animals are no longer making insulin. These animals are completely diabetic. So now you can find a way to try to repair. So in that study, they injected streptozotocin plus endogenous stem cell mobilization, the release of the animal's own stem cells. And you can see the partial reconstitution, but this is enough to make it so that these animals are no longer diabetic. You don't need 100% of function of your pancreas to not have the symptoms. So these animals have been normalized. Very similar study has been done in 14 humans recently diagnosed for insulin-dependent diabetes. They did 2 rounds of stem cell release. Of the 14, 13 were independent from insulin between 3 to 36 months. I'm not telling you we've discovered a treatment for diabetes. I just want to show you what the stem cells in your body have been documented to have the ability to do. They can become many things in your body. They do become many things in your body. They've done this from the day you were born. Let's look at the last one, from the nervous system. I'm taking systems or aspects of the body that have been fairly well known to not have the ability to regenerate. When I was in school, the heart did not regenerate. The brain, my background is neurophysiology, brain research. I was told, we don't make new brain cells. You're born with what you have and good luck. You don't make new brain cells. This is completely overcome right now. We know very well that we make new brain cells, but it's just new science. So here's a study on Parkinson. So for Parkinson, what they do is that they inject a toxin called 6-hydroxydopamine, and it's injected in an area of the brain called the substantia nigra. Forget these words, they're not relevant. It's the part of the brain in a Parkinson patient, the part that is dead. There's dopamine cell in that area, and when the dopamine cell dies, you develop the symptoms of what is called Parkinson. So if you inject 6-hydroxydopamine in that area, it kills dopamine cells, and then the mice will develop Parkinson. The way that they quantify Parkinson in animals is that their lack of coordination makes them reluctant. They hesitate before moving. So a normal mouse will stay immobile in its cage about 10 seconds. So you've all seen mice, they walk, they stop, they walk, they stop. They stop about 10 seconds. But once they have developed Parkinson, it's more than a minute, where they hesitate to move because of lack of coordination. If you give them the normal treatment for humans, L-Dopa, carbidopa, you've got a very nice improvement. But if you trigger the release of the mouse own stem cells, you're almost like back to control. Now, to quantify the progression of the loss of coordination, the scientists did something very ingenious. They put a piece of tape on the nose of the mouse, and they counted the time to remove it. Sometimes you don't have to be sophisticated to quantify something. So in the control group, so this is 1 day, 1 week, 2 weeks. In the control group, you see that there's almost like a learning that takes place. They're doing it very quick. In the group that developed, the control Parkinson group, you can see the evolution of the loss of coordination. After 2 weeks, about 25 seconds to remove the piece of tape. With L-Dopa, carbidopa, same evolution, just slower. In the group in which we triggered the release of the mouse own stem cells, same decline, and then after that, reversal. And that's the kind of things that has been documented also in humans. I won't go into this data, but so just a few studies just to illustrate here that if you can increase the number of stem cells in circulation, yes, the body has the ability of using these stem cells, and they will migrate where you need tissue repair. The third conclusion is, to me, so far so good?
Yes.
Too technical? No? Okay, Canadians are smart. So, okay, the third conclusion, fewer stem cells in circulation has been linked to loss of health. That's true. That's true. So, fewer stem cells has been linked to developing health problems. So, which is the corollary of these two, which means if you have more, you feel better, and this one is if you have less, then you're at risk for not feeling better. So, and that is what I mean when I say this is called to really change the way that we look at health and wellness, and even the way that we practice medicine. So, let's spend a little bit more time here on this one. So, it comes from the fact, Martine, could you bring me some water or somebody? Thanks. Okay, thanks. So, it comes from the observation. Remember the very, very first study that I talked about when scientists studied leukemia? So they irradiated female mice and then they injected male stem cells. They saw that it worked, but they found a brain cell with the Y chromosome. These mice did not have traumatic brain injury. And yet, we found a brain cell with the Y chromosome. What I mean here is that while stem cells will... Thank you. While stem cells will primarily go to a site of injury, they will also, to a lesser extent in the background, also go to tissues where you do not have injury in a normal process of tissue renewal, tissue turnover. So you lose cells every day and you replace them every day. So let me show one study here. Oh, before I go there, yeah. So before I go there, let's just talk about this. Because this is all put into the context of the current understanding of disease formation. What is a disease? A disease is, we don't say it that way, but if you just think about it 30 seconds, all diseases are, okay, cellular loss. You lose cells in your heart, you have cardiomyopathy. You lose cells in your liver, you got liver problem. You lose cells in your lung, emphysema. All the problems linked to the lung is the loss of cells or function of the cells. You lose cells making insulin, you've got diabetes. You lose cells making dopamine of your brain, Parkinson. Think of any degenerative disease, it is always, without an exception, the loss of cells of a certain tissue. You lose the cells making the myelin around your nerves, you've got multiple sclerosis. Every disease is always the loss of a cell or a function of a cell. And the general understanding is that you're born, you mature, then you reach 20, 25 years of age, you've reached optimal health, and then from then it's a slow decline as you lose cells, and where you're going to lose cells most, this is where you will express some sort of health problems. Fine enough?
Yes.
This is the general understanding of health. But what we're discovering with stem cell research is that, remember again this study on leukemia, when we saw stem cells becoming brain cells, is that it's also happening to a slower degree in every single organs and tissue of the body. So let's look at one study. It's the only study that I know of that has studied the phenomenon. The study I talked about before on leukemia, they studied leukemia and then discovered this in the brain. Other studies have studied, for example, heart attack, and then they found cells in the lung, in the liver. So many observations have been made in various studies, and at one point, one group decided to really study the phenomenon. And they did this in a very ingenious way. So it's a little bit technical, but follow me at the end, the conclusion is simple. In nature, is it too technical or it's fine so far?
Fine.
Good, okay. So, in the environment, you've got, historically, we have 0 radioactive carbon. But after the beginning of nuclear testing, in the 1950s, there was a very sharp rise in the concentration of radioactive carbon in the atmosphere. Until 1965, when we had the international ban of above-ground nuclear testing, and then we've got this slow diffusion of radioactive carbon through the stratosphere. Okay? Now, carbon in the atmosphere is present in the form of CO2, which is incorporated into plant through photosynthesis. The plant are eaten by the animals that we eat, or we eat directly the plant. Conclusion, the amount of radioactive carbon that I find in my body, in my diet, reflects from a few weeks to a few months, the concentration of radioactive carbon in the atmosphere. So the day that one of my cells divides, and the DNA divides, and I need to duplicate the DNA, the new filament of DNA is made of carbon. Where is that carbon coming from? From my diet. So the ratio of radioactive carbon in the DNA of a cell will reflect the ratio that was in my diet that reflects what was in the atmosphere. So if I take cells from an individual born after 1965, I can date the birth of that cell by simply quantifying the ratio of radioactive carbon. And that can tell me when that cell was born. So now it allows me to test 2 opposing views. The first view is that I mature, and the traditional view is that after about 5-6 years of age, I have all the cells in my heart that I will have, and from then on, it's just a slow decline. Same thing in the brain, same thing in various tissues. Or I can see that throughout the entire life of an individual, I have new cells that are showing up. So it's more a process of losing cells and replacing them. Conclusion of that study, we have about half of a new heart every 25 years. Half of a new liver every 2-3 years. A new pancreas every 4-5 years. A new lung every so many years. The time frame here is not relevant because it has been rarely studied to establish a time frame. But out of what has been observed, everything is in a constant process of turnover. So really health is not a slow decline. Health is a balance between 2 phenomena. Losing cells and replacing them. When you're young, you've got a lot of stem cells. So you repair very quickly. Look at kids, they get injured and they just repair so quickly. But as we age, the red marrow that makes stem cells transforms into yellow marrow, fatty marrow, that does not make stem cells. So we have fewer and fewer stem cells until the day where we switch the balance, and now we have more cellular loss than we have renewal. It's a little bit like saying you're born with a bank account. So the 2 views is, one, you're born with a bank account with a certain amount of money, and I tell you, spend it wisely. Or I tell you, all your life you will have an income. As long as it fits your expenses, you're fine. And obviously, it makes so much more sense that way. What will make you develop a problem or another, why somebody has Parkinson, while somebody else is emphysema, diabetes, whatever. It's a whole series of factors. It's your genetics, your past injuries, your exposure to environmental toxin, your lifestyle, your exposure to physical stress, emotional stress, your diet, your ability to rest, your mother-in-law. All of these factors, they are the pressure applied on you and your ability to respond to it. We've all seen this. There's nothing here that you're learning. You know all of this. We've all seen this in our life. Two individuals with the same challenge in their life. One will just go through it without a problem. The other one is crushed. You know, we all made of a whole set of different problems, which will dictate where the problem shows up. Let me make another banking analogy, just to understand what I mean here. It's like saying, I've got somebody who cannot pay for his car, somebody else cannot pay for their mortgage, somebody else cannot pay for their schooling, and somebody else cannot pay for their health bill. So you have car problem, house problem, school problem, and health problems. But I really just have cash problems. It's the only thing I have. It shows up differently in the life of different people. Here I've got one problem. Not enough stem cells in the bloodstream to compensate for cellular loss. And it shows up in different ways because of different circumstances, and I give it a different label. If it's in the pancreas, I say diabetes. If it's in the heart, I say cardiac patient. If it's in the brain, I call it Parkinson. The bottom line is just fewer stem cells in circulation. There's a way to test for that if this is true. And the way to test for this is to count the number of stem cells in the bloodstream of people who have developed different kinds of health problems. So we proposed this about 10 years ago, and at the time, what I was saying is that it would be very interesting to go and quantify how many stem cells we have in the bloodstream of people who have developed various types of degenerative health problems. Across the board, statistically, what we should find is that overall, people who've developed various problems should have fewer stem cells. It's obvious now with what we've talked about, but it needs to be tested. Now there are many studies that have done exactly that. Here's one, where the scientists quantified the number of stem cells in the bloodstream of people with normal glucose tolerance, impaired fasting glucose, impaired glucose tolerance, and diabetes. And you can see there's a straight line. People that are in advancing or progressive stages of the development of the problem have fewer and fewer stem cells. Kidney function. Here are, oh sorry, I clicked the laser. So this is the kidney function in healthy controls. The red line is the average in healthy control, and in people of declining kidney function. And you can see here less than half the number of stem cells in people who have developed kidney problem. If you plot number of stem cells with kidney function, you get a straight line. There's a direct relationship with kidney function and number of stem cells in circulation. Next one, patient to complain of erectile dysfunction and control patient. If I bring the averages to the axis here, you can see people who complain of erectile dysfunction have less than half, a little bit less than half, the number of stem cells found in healthy people. If you take all these people and you establish their international index of erectile function and you plot that with the stem cell, you get a straight line. There's a direct relationship between how many stem cells and erectile function. Erectile function has a lot to do with vascular function. The same thing has been seen with vascular health. This is a study in which they quantified the thickness of arteries. And that's a way to quantify the development of developing atherosclerosis. So the health of your cardiovascular system. So as people have higher thickness, you can see they also have fewer stem cells. When you do the relationship, straight relationship. Same thing with hypertension. People with hypertension, fewer stem cells in circulation. Forget the disease and all of this. The disease are the tools that we have, the studies, the things we investigate to be able to expose the phenomenon. The point here is simply people who have fewer stem cells in circulation, the cells that are lost are not replaced properly, so these tissues start to function less than optimally. It's just that simple. Here's another one with digestive problems, Crohn's disease, colitis, less than half the number of stem cells found in healthy people. The last one, lung. This is lung problem, number of stem cells. In that study, they did not only do lung, they also studied the function, their ability to adhere to the capillary wall before migrating in the tissue, so everything is reduced. Most of the time when they study more than just the number, they find also the same kind of relationship. And that's the last one. This is joint problem. Same thing, people with joint problem, fewer stem cells in circulation. Forget now all the diseases. They're just the ways that we have to expose the phenomenon and document the process in the body. The conclusion is super simple. If you have more stem cells in circulation, better stem cell function, your body is better capable day to day to just maintain itself, and it just adds up to very impressive results over time. It's really that simple. So now let's put aside all disease, keep just that in mind. If you have more stem cells in circulation, your body is just more capable of maintaining its own health. And that's where RevitalBlu comes into play. So RevitalBlu is not going to reverse diabetes, heart, whatever. It's not going to do any of this. It does one little simple thing. It helps the body to put a little bit more stem cells in circulation. It's a little bit like if I'm telling you food is not a miracle. But if you cannot eat and I give you just a little bit more food every day, you can make a whole lot of a difference in your life. That's all we mean here. We just give back and hence the natural ability of your body to do what your body is born to do. So when I started all of this 18, 19 years ago, I was working with this plant. It's an aquatic botanical, It's an aquatic botanical, a blue-green algae, and it grows naturally in a lake in Klamath Falls, Oregon. And I was hired at the time to study the health. How much time do I have, Martine? Do you know how much time I have left?
About 5 more minutes.
5 more minutes? Okay. So, I need to wrap up quickly. Okay, so I started to study this plant at the time when regulation had just been passed in the United States, forcing companies selling a product to document the claims that they would have about a product. So that product was used for inflammation, to support immunity, and also people were feeling like better mental clarity with this product, just feeling mood elevation. So we documented all of these with proper molecules, active compounds, and all of this in AFA. But in the course of these studies, we also came across cases that were very impressive, that touch various aspects of human health. Very significant improvement in cardiac function in some people, pancreatic function in others, lung function in others, liver function, brain function, the retina, the joints, the skin. So the question was, what is it that this product does that seems to touch so many aspects of human health? We did not have a response until a colleague of mine sent me an article. The title was Turning Blood into Brain. It's that first study that I told you about. So when I looked at that, it started to trigger this process. Not the first time, the second time I read it. I started to think, well, wait a minute. Have we seen this in other tissues? So at the time, 2000, 2001, it had already been reported in the heart and in the liver. So we started to think of this whole concept about stem cells. So the idea was twofold. On one hand, what's the natural role of stem cells in the body? And if it turns out to be your repair system, second question, what if that plant actually worked by supporting the body's ability to release stem cells? It would then explain why so many different people had different kinds of benefits. So we took about a year to develop the protocol, we tested it, and then the discovery is that an hour after consumption, indeed, we have more stem cells in circulation. Do you understand why that means? Just to explain the protocol very briefly. So people come in the morning around 8, we let them sit so that the blood sort of normalizes, they're resting, because movement, stress can affect stem cells, or at least our ability to quantify them. So they rest for an hour, then we do a first blood draw, then we give them placebo or test product, and then we take other blood samples after an hour, 2 hours, 3 hours, and then we pass these blood samples into a machine called a flow cytometer. It lines up all the blood cells one by one in a very fine tube, and then it sheds a laser on them with a photoelectric plate on the other side, and it allows to basically identify what's that cell that just passed, and it counts stem cells. It's the method used everywhere to count stem cells. So using this method, we can show that as times go by, after 1 hour, we've got an increase of about 25 to 30% in the number of circulating stem cells, and it comes back to control after 3 hours. So after documenting this plant, it was clear to me there cannot be just one plant having an effect on stem cells in nature. If we have evolved in symbiosis with the environment, there has to be other plants that have an effect on stem cells. So the question was, what else is known out there historically to be associated to a broad variety of health benefits? Medicinal mushroom, goji berry, or longevity, goji berry, things that you've all heard about. So we tested them, and we found, very interestingly, virtually all of them have an effect on stem cells to some extent. But through all of these that we studied, the next one that to me was very interesting was a berry that comes from Tibet called sea buckthorn berry. Sea buckthorn is very interesting because I asked a colleague from China years ago, I said, okay, tell me from Chinese medicine, if I'm lost on a desert island and there's only one thing that I can use, what would it be? She said, sea buckthorn. I just thought, how can it be? Just a berry? So it took me a lot of time to go to the scientific literature and get the motivation to actually test it. And then I discovered that there's actually a very interesting history behind this berry. In science, most plants are called according to what they look like. So if you look at Aphanizomenon flassaqua, it means invisible flower of water. And most plants are named according to physical characteristics, except, sorry, click on the wrong button. The name for sea buckthorn is Hippophae, which means horse light. It means shining horse. It's Alexander the Great traveling into Western China during his conquest, and at some point he abandoned a herd of horses wounded in battle, and he left them to die in a field of sea buckthorn. And a few weeks later, he came back and he saw a herd of vibrant, healthy horses with a shiny coat. If you know horses, a shiny coat means health for a horse. So the berry was called shining horse. From there, the same observation by Genghis Khan, from there it got incorporated into Chinese medicine, Mongolian medicine, Tibetan medicine, where it has been used for problems of the heart, overall cardiovascular system, pancreatic function, lung function, injuries, cut, wounds, burns. So think about everything we've talked about. When I read this in the literature, to me it tells me there's a good chance here that I have an effect on stem cells. So we just tested it, and we developed an extract from the sea buckthorn berry, and we saw over time a different dynamics. It's just not a release that comes back to control, but a slowly increasing number of stem cells in circulation that last a little bit longer than what we had with AFA. So we have other ingredients. I will go directly to the formula. So these different ingredients are all active compounds, or I should say plants, that have been documented to support stem cell function in the body, their release or their migration into tissue. So what RevitaBlu does in the body is, according to everything that we've said right now, you will understand, support the body's natural renewal system. The AFA and the beta-glucan supports immune function. The sea buckthorn and the aloe supports provide antioxidant protection. All of these adds up to healthy aging. And there's some biochemistry in the product. I won't have the time to go through it, but that will provide the brain with molecules that will give you the impression of just mental clarity, sort of a mental energy, mood elevation. You'll just feel better. And it is probably so far the comment that I've heard the most about people taking RevitaBlu. They take it and they just feel like they're more there, they're more aware. There's a chemistry, it's not illusion. Okay, when we develop this, I'm almost done. When we develop, is it too much or is it okay?
No, that is
okay. So until what, 3, 4? Okay. So just when we develop, it's the last part. When I started to develop RevitaBlu, Dr. Entar, she's in charge of product development at Jeunesse, and she said, as you develop RevitaBlu, make sure you go and really study the current products that we have, because you will see that there's a very nice synergy with these products. So I studied the products, and first observation or conclusion is that understand here that you have really, really top quality products at Jeunesse. And what I mean there is that you can have two products that have on their label the same name as ingredients, but there can be vast differences between these ingredients, and you don't know that as a consumer until you start to develop product, get quotes from manufacturers, and test these ingredients. You don't understand the vast difference in quality that you can get. Let me just give you a quick example. I worked a lot with fucoidan in the past. It's a very complex polysaccharide with a lot of effect on stem cells, but also on inflammation, on cancer. So a very interesting compound. You can buy a good fucoidan for about $1,000 a kilo, but I can also find it at $200 a kilo. What's the difference? I mean, why would I pay $1,000 if I can pay $200, right? So I got a lot of these $200, and then we tested them. We use HPLC. It's a standard method to test molecules. On HPLC, they're both the same. They're both fucoidan. But if you dig a little bit deeper, fucoidan, fuco means fucose. It's a polysaccharide that is made of at least 30% fucose, about 2% glucose, about 10% sulfate. We tested the other fucoidan. It was 85% glucose, 0.2% fucose, and about 1% sulfate. It is starch made by a brilliant team in China to make it look like fucoidan. And when you taste it, it tastes like flour. So it does taste like what starch would taste. But it's sold to you at a good price, $200 instead of $1,000, but it has none of the benefit. But if you buy it because it's cheap and it gives you better margin, you put it in your product, you've got the right, because you have the certificate of analysis, to say you have fucoidan in your product. So I'm just saying this here because I looked at them. These are good quality products, but there's a nice synergy between Revitable and these products. What I mean by synergy, I do not mean we went in the lab and we studied the synergy. What I mean here is that if I develop Revitable for a company, and then they come and they say, what would be your next best product to support what Revitable would do? I would say Reserve. Because, I didn't go into the detail of this, but when you release stem cells, there is something that can reduce the ability of stem cells to find in the body what are the tissues that are in need of repair. And it is systemic inflammation and oxidative stress. So if you can suppress and decrease oxidative stress in the body with strong antioxidant, then you can assist the role of stem cells. So let's say you release, hypothetically here, you release 5 million stem cells with 1 sachet of Revitable. If of these 5 million, 1 million reach your tissue, you get 1 million cells worth of tissue repair. But if you allow 5 million stem cells to reach the tissue, then you've got much better repair. That's what we're talking about. Supporting the ability of stem cells to circulate and go where they need to go supports the whole process. And resveratrol in Reserve has also been documented to support proliferation of stem cells in the bone marrow. Now, next one, Luminesse. Very interesting combination here. To understand how that synergy works, we just need to understand what Luminesse really does. I know that the lady that came before, Yibin, she did a wonderful presentation. She summarized Revitable in a few lines in a very, very good way. So, is she here? Good job. She did very good. And she also described Luminesse. So let's go a little bit deeper in Luminesse. Luminesse is a product designed, developed to tap into the very natural process that the skin uses to regenerate itself and renew itself. So there's a layer in the skin, it's a third layer, and it contains stem cells that are called epidermal stem cells. These epidermal stem cells are not immortal. They multiply for a certain number of generations and then they're no more. So they need to be replenished. So they come from the blood, which comes from the bone marrow. So you get bone marrow to the blood to the skin they become skin cells, they migrate to the surface and they flake off. And you've got this constant renewal of your skin every week or two. So you don't realize it, but you have so much renewal and regeneration taking place in your skin from the day you are born. So this product is made out of peptides that have been shown, demonstrated, to basically help attract stem cells from the blood to the skin, help them proliferate and become cells of the skin. No miracles here and nothing that is exceptional. It just supports this very natural process in the skin. Now, if you put stem cells in the blood, they will migrate to the skin. It's not rare to see somebody taking Revita-Blu, and 2 months later they will say, many people tell me, what are you doing? You look more yet radiant, you look younger. Because they go to the skin as well, so they can change what your skin looks like. But if you apply peptides on top of your skin, the serum will do its same effect, but now there's more stem cells in the blood to attract. So you amplify the process. You use both products separately, you get benefits. You use them together, you just enhance the benefits that you can get. And you've got like this, I won't go too far, you've got that same synergy with almost every product. AM, PM, when we realize that one stem cell can become about 1 to 5,000 tissue cells, that means you release 5 million, you've got 5 to 25 billion tissue cells. So we realize it's a very demanding process for the body. There's a significant metabolic demand. So you need all the vitamins, the minerals that the body needs. As these cells multiply, you need to also protect DNA in this process, and that's a lot what AM/PM was developed to do. As cells multiply, they need all the amino acids that the body needs. You get that with Zen. And the last one, which is also very interesting to talk about, it's Reveal. Reveal is to the scalp what Luminous is to the skin. It's that simple. On top of having epidermal stem cells, in the scalp, you have hair follicle stem cells, melanocyte stem cells. With age, in some people, they get dormant, they don't renew, so you lose your hair and they don't come back. So if you can reawaken that layer by attracting stem cells and making them functional in the skin, you do no miracle here. You just give back the skin its natural function of making hair. It's really that simple. So that's what I wanted to share with you. I hope it was not too technical or too complicated. The message at the end is super simple. Yibing mentioned it very simply. Stem cells are your natural repair, renewal system. More stem cells in circulation, your body can maintain itself better. So give it a try. The product is not going to cure anything. Can the product cure this? Can it do this in such and such disease? There's no answer to that question. The only answer is, I don't know. Give it a try and see what it can do for you. What I can tell you is that stem cells can become liver cells, heart cells, pancreatic cells, brain cells, lung cells, liver cells, brain cells, retinal cells. They can become anything. Your body will decide where they go and what they become. So just take the product, give it a try, and see what it does for you. Okay? I don't know if we have time for questions. We have time for one question. I see 2 hands there, so let's go. First.
Okay, I just, what the lady said before, that you have to have hair follicles. You can't lose all your hair and expect it to come back, right? So in the case of men,
Yeah.
especially, or women too, before they start getting really bald, would it help?
Of course. That's exactly at the heart of what we're talking about.
Well, that's
Yeah,
what
yeah.
I'm hearing.
Yeah.
That's what I'm hearing. That's what I'm
That's
hearing.
the whole concept behind the Reveal and combining it, is that yes, this can happen. I mean, I don't want here to make claims, but let me just share a funny story. In the company where we launched the very first product that I developed some 14 years ago, there was this fellow. We saw it in many people, but there was this fellow. His business card was that he was balding, all his hair was white, but after taking the product, his crown was black. So he looked like a skunk. But he didn't do anything because everybody would ask him what's happening, and he would then talk about the product. So after seeing enough people like this, we just thought, first people would tell me it's changing my hair color, and I would literally tell people, stop making up stories. You know, the product is good enough on its own. Like, you can't change your color. You know, I mean, it was really upsetting me. And then one day a friend of mine says, no, it's happening to me. So we went into a lab of cosmetology and we measured the lab measured the density of white in the hair, and after 6 months, we saw in every single participant in the study about 20% reduction in white, because stem cells can become melanocyte stem cells. In that study, we did not quantify hair growth, but we saw it in a number of people. So absolutely, here the mechanism of action is that where stem cells can migrate in the scalp and become functional hair follicle, then it's very possible that it happens. I cannot tell you that it will happen, but that's the mechanism.
What about estrogen in women? Does it do anything?
Stems, okay, let me use that question to answer all the questions that are going to be My mother, my father, my uncle, my neighbor, my son, as whatever, my parrots, my dog, my lizard, whatever, as whatever problem, can the product help? No, I cannot tell you that it will help because I have not done any specific studies with a disease. But I can tell you, stem cells can become cells of the thyroid gland, of the pancreas, of the liver, of the lung, of the brain. Everything regenerates in the body with stem cells. There's documents for every single tissue. So I can tell you where these stem cells will. The product won't do it. The product just put more stem cells in circulation, your own stem cells, and they will go wherever your physiology will lead them.
Okay.
So you give it a try and you tell me next time Martine invites me to Montreal.
I'm sorry, we're so behind with the program.
It's important. As you said, you don't know if it can help us, but on the flip side, can it harm us in any way?
No.
Especially if you've had like some serious disease like a cancer or something. Or
No.
whatever. Because doctors scare you into believing
Yeah.
that you shouldn't try it.
No, it's safe.
Okay.
I'm so sorry. This is the smartest person I know, and I know that you enjoy him just as much as I do. Unfortunately, we are running out of time, and we also have another amazing speaker.